Hi everyone, in this video we're going to talk about the amino acids. Amino acids are the building blocks of proteins. There are 20 amino acids, naturally occurring amino acids. And as we'll see, they're going to have sort of a generalized structure, all 20 different amino acids, but they'll differ in one important respect. So if we were to draw out a sort of a generic amino acid, they have this structure. Now notice that I've put some stereochemistry here. I've put sort of a wedge bond here for this hydrogen and a dash for the amino group. We'll talk more about stereochemistry a little later, but it turns out all of the amino acids have this particular stereochemistry. This is called the L stereochemistry. So all of the amino acids are called L amino acids. And as the name would indicate, there is an amino group, which is shown here, and a carboxylic acid. So we have an amino and an acid. And then notice I've put an R group here. The R group is referred to as the side chain. And it's this side chain that's going to be different between the 20 different amino acids. So here we have, again, our carboxylic acid. Okay. Recall that when we talk about carboxylic acids, if you look at a carbon that's adjacent to a carboxylic acid, this is called an alpha carbon. So this group here is sometimes referred to as the alpha amino group. Okay, because it's coming off of the alpha carbon. And then this would be the so-called alpha hydrogen. So again, this is sort of the generic structure of all the amino acids. And because it has both a carboxylic acid and an amine, this can act as either an acid or a base. So let's just draw this a little simpler now. I won't worry about the stereochemistry anymore. Just draw it like this. So if you were to just draw a generic amino acid, not worry about what pH this is or its protonation state, you might draw it like this. However, at physiological pH, so at pH approximately 7.4, which is physiological pH, this amino group and this carboxylic acid will exist in a different protonation state. So what's going to happen is that the amino group, since it's a base, the typical pKa for the ammonium ion of this is going to be up around 9 to 9.5. This is going to not exist as the NH2, it's going to exist as an NH3. Okay, so at physiological pH, the amino group will become protonated and have a positive charge. Conversely, the carboxylic acid, which typically has a pKa around somewhere between 2 and 5, at pH 7.4, this proton will not be there, and this will exist actually as the carboxylate anion. So amino acids, all 20 of them, have this general structure at physiological pH. And when it comes to sort of memorizing the amino acid structure, I would tend to say, let's try and memorize them so that we know what the protonation state is at physiological pH. All right? One final thing I'll mention is that this type of species where you have both a plus and a minus charge is called a Whitter Ion. Okay, it's basically an old German word that just means a double ion. So all of the amino acids exist as Whitter ions at physiological pH. So this is the sort of generic structure of those. Now in the next section we'll talk about what are the different R groups and how can we then group the amino acids into different sort of classes. So when it comes to grouping the amino acids, what you want to think about is the sort of characteristic properties of the side chains or the R group that we talked about in those. So what I've drawn here are 5 of the amino acids. And I've just abbreviated the side chain. So I've sort of cut off the rest of the molecule, the alpha carbon, the carboxylic acid, the amino, and I'm just focusing on the side chain. So if my side chain is an H, this is called glycine, abbreviated GLY, okay, a three-letter abbreviation. And it's classified as a nonpolar amino acid. Okay, it doesn't have any functional groups really in it, just a hydrogen. If we add, say, a methyl group to this, instead of a hydrogen, this becomes alanine. ALA would be the abbreviation. Next, notice we have an isopropyl group here, so a 3-carbon group. This becomes valine, VAL. Next, we have sort of a sec-butyl group that would be attached. This is called isoleucine, or ILE. Next, we have this group here, which is similar to isoleucine, but notice we've moved the methyl group out one position. This is called leucine, or LEU. So you can see in this series that we're essentially adding more and more carbons, making the polarity of these groups go down. So this becomes more nonpolar as we go down this series. All right, so these 5 amino acids, 5 of the 20, can be classified as nonpolar amino acids. Now there are a couple others. I'll just briefly draw those out. If I have something that looks like this, now I have a CH2 and a benzene, that's called phenylalanine. Or PHE. Right, so that definitely is a very nonpolar group. It has this benzene-like molecule in there. Those are a couple of the others. There's one other one that's usually classified as a nonpolar. This one actually has a sulfur in it, but we'll just, it actually has an ether. Okay, so this thioether or sulfide type group, notice it's got sort of long carbon chain, sulfur, then the methyl group. This is called methionine, or MET, M-E-T. Now, even though it's got the sulfur, it's still more like an ether or a thioether, and those tend to be very nonpolar as well. So collectively, those 7 amino acids would be called the nonpolar amino acids. So let's look at these sort of in order. The first one here has a CH2 and an OH, so just a simple primary alcohol. This is called serine, or SER. The next one you notice has actually a secondary alcohol. This is called threonine, THR. Next, we have something that's very similar to serine, except now we're replacing the alcohol with a sulfhydride or a thiol group. This is called cysteine, or CYS. Next, we have a primary amide. This one that has a CH2 and then the primary amide. This is called asparagine, or ASN. The next one you see is very similar to asparagine. It's just got an extra CH2 in its side chain. This is called glutamine, or GLN. And then lastly, we have something that's kind of like the phenylalanine we saw in the previous group, except now we've put on an OH group. So this is a phenol, which makes this a much more polar molecule than the regular benzene molecule. This is called tyrosine. So collectively, all of these groups contain functional groups that can hydrogen bond to water. And what we'll tend to see is when these are in protein structures, these will tend to hydrogen bond with the water that surrounds a folded protein. The prior groups, the nonpolar groups, they tend to be sort of buried more into the middle of a protein. The next grouping is called the acidic side chains. And these functional groups, or these side chains, will contain carboxylic acid functional groups. Now, I've drawn these in the protonated form. However, recall that at physiological pH 7.4, these carboxylic acids are not going to exist like this. So we're going to draw the the carboxylate form of these. Okay, so this carboxylic acid will be converted into this. And so this shorter carboxylic acid, this is called aspartic acid. ASP. And the carboxylate form is just called aspartate. All right, so the protonated form we call it aspartic acid. This would be called aspartate. This little bit longer carboxylic acid, this also would not exist with the proton on there. It's going to look like this. Is the carboxylate at physiological pH. This is called glutamic acid. Or GLU. So the actual carboxylic acid is called glutamic acid. The carboxylate would be technically called glutamate. So that covers the acidic side chains. The next section, we'll look at the basic side chains. So what I've drawn here are what are called the basic side chains. These are side chains that all contain nitrogenous bases. Now I've gone ahead, just for simplicity, I've drawn these out as they would appear at physiological pH. So this first one has a long 4-carbon chain and terminates in amine. But I've drawn the amine as the ammonium ion, because that's what it would look like at physiological pH. This is called lysine. Okay, or LYS. This next one has an interesting 5-membered ring structure. This ring is called an indole, or I'm sorry, not an indole, an imidazole ring. So notice there's actually 2 nitrogens in there, but one of them acts as a decent base and will become protonated. This is called histidine, or his. I'll just mention that the pKa of these is around the pKa of neutral pH, around 7, and so you'll often find histidines in the active sites of enzymes. Lastly, I've drawn this molecule that has a 3-carbon chain, and then this kind of unusual 3-nitrogen containing compound. This group, I'll just put a little sort of dotted line around it, is a protonated amine, so it's got a positively charged. This is called a guanidino group. Okay, but the overall amino acid is called arginine, or ARG. This is the pirate's favorite amino acid, arginine. All right, in the last little section, I've just got 2 more to cover. These are sort of the unusual. There's 2 sort of unusual amino acids that are kind of hard to classify. Okay, the last 2 amino acids of the 20 that I wanted to draw out sort of separately are sort of the red-headed stepchildren of amino acids. They're kind of hard to classify. This first one is the only cyclic amino acid. So you'll notice coming off the alpha carbon, there's this chain, but it sort of comes around and then reconnects to the nitrogen. Now I've shown this at physiological pH, so this nitrogen would have 4 bonds, would have a plus charge on it, and the carboxylic acid would be the carboxylate. This is proline, or PRO. It's the only cyclic amino acid. And you often find proline in proteins where the protein might be going in one direction and they need to make a bend or a turn. You often find prolines in turns of protein structures. The last one has a side chain that looks kind of like some of the other ones we've seen, but you'll notice now it's got sort of a benzene ring, but it also has this 5-membered nitrogenous ring. This is the amino acid called tryptophan, or TRP. Technically, this really is a fairly non-polar amino acid. But because it has this nitrogen in there, sometimes people classify it as polar. You also don't, it's not very basic either. This nitrogen is not very basic. So it's kind of hard to classify in one of those groupings. This is also the rarest amino acid in proteins. You typically only find a couple of them in proteins. And the last thing I'll mention is that this ring system here, this 5-membered nitrogenous ring and the benzene ring. This is called an indole ring. And that compound is ultimately metabolized into a number of different neurotransmitters, and also a number of different drugs have this indole type ring system in it. All right, so that kind of covers the structure of the amino acids, and then we've kind of broken down the 20 different amino acid side chains. In general, you should become very familiar with the side chains and certainly the three-letter abbreviations and the full names. And then certainly understand what the protonation states of the carboxylic acid and the amines would be. In general, you'll often be sort of given these side chains sort of as a cheat sheet on an exam, but in some classes you might be expected to actually memorize all 20 amino acid side chains. All right, so I hope that helps out with amino acids. Good luck.